Bismillahirrahmanirrahim, and welcome back to this next video. And this video is the continuation of the previous video where I told you how to use the genetic code table. In my previous video, I've told you that from a given starting point in your DNA sequence, if you start reading the sequence like 3 nucleotides or 1 codon at a time, then you consult the genetic code table that is going to give you the corresponding amino acid sequence of your DNA sequence. And I've told you, say for example, if this is the sequence, so the first thing you are going to read your sequence, then you are going to decompose that into the successive triplets. By that I mean you have to make the codons, and then you consult the genetic code table, and the genetic code table is going to give you the sequence of the amino acid from the given sequence of the codons. And this was one of the example that I discussed with you. And then I've told you that if your DNA sequence is correctly listed in the 5' to 3' orientation, you actually generate a sequence that is in the conventional end to C terminal as well. Then important thing to keep in mind that I want to focus on this video is that if you know where a protein coding region starts in a DNA sequence, this is the most important thing, that you should know where a protein coding region starts in your DNA. Your computers can pretend to be a cell, and it can actually give you the corresponding amino acid sequence of the DNA that you have given to it. So this simple computer translation exercise that is actually at the origin of most of the so-called protein sequences that you can find in different protein databases that are available on the internet. So many sequence analysis programs, they actually acknowledge this fact that if you give them a DNA sequence, they are going to convert them into the triplets, and they are going to give you the corresponding amino acid sequence, and they actually offer this on the fly translation, so you can process your DNA sequence as virtual protein sequence with a simple mouse click. So as I've told you that the important thing is that where to start making the codons, or from where to start to making the triplets. That is very important. So once you know that where the protein sequence is going to start, then you use the genetic code table to give you the you can say desired or the required DNA protein sequence. See, for example, this was one of the example that I discussed in my previous video. So if this is the messenger RNA sequence, as I told you in my previous video, don't worry about the T's because in the messenger RNA you usually do not see any T, but this T, the programs, they usually translate them or they usually read them as the U sequence. So if this is the sequence of the messenger RNA, this is going to be the resulting amino acid sequence that you will get. But if you look at this, what you are going to do is you are starting from the this particular end. So the first codon, that is ATG, the second one is then the GAA, the third one is the GTA, and so on. And if you have made these codons, you are going to get a desired sequence. For example, the first in this particular is the ATG. So the first amino acid you are going to get is the methionine, and so on. But if you are going to change the amino acid sequence like into a different one, as particularly at this one, as you can see, you're not starting from the A, you are starting from the T. So if you start from the T, that means that the first codon that you are going to get, that is the TGG. That means that the second codon you are going to get is the AAG. So if you skip the first nucleotide, the codon sequence that you are getting, that is totally different than the one when you start from the nucleotide A. So if you look at this particular, this is the sequence. The sequence of the nucleotide remains the same. The difference is from where you are going to start the codon. From where you are going to start the or making the triplets. So if you look at this particular sequence, the amino acid sequence that you are going to get is, the first one would be the tryptophan, the next one would be the lysine, the third one would be the tyrosine, the fourth one will be the leucine, and so on. What this means is that it is very important to identify where to start making the codons, because if you are making a different codons, you are going to get a very different kind of the amino acid sequence. So like for example, if you start from the third position, like from the G over here, you will be getting the GAA and so on. That means again you will be getting a very different kind of the protein. So it's important to identify the starting point in your DNA sequence and making the codons, because if you are making the codons from a different position, the amino acid sequence that you are going to get will be totally different. We will continue this discussion in the next video.